Hello everybody, let us see some of the questions from second part of molecular basis of inheritance. Right away, let us enter into the first question. Choose the incorrect match. Frederick Griffith, Streptococcus pneumoniae. This is the correct match. Oswald Avery, bacteriophage. This is the incorrect one. Because Oswald Avery carried out experiments, Avery, MacLeod and McCarthy carried out experiments the same bacterium, pneumococcus or streptococcus pneumoniae, but not bacteriophages. Martha Chase, Martha Chase and Alfred Hershey, they carried out experiments by using T2 bacteriophage and Escherichia coli. The second option is the incorrect one. So answer must be the true. Let us see the answer. Come to the solution. Oswald Avery, McLean McCarthy, and Colin MacLeod isolated biochemicals from a strain of pneumococcus and established that DNA as genetic material obtained by R strain from heat-killed S strain by using the enzymes, proteases, RNases, and DNases. They have not worked with bacteriophages. Frederick Griffith worked with R and S strains of Streptococcus pneumoniae and showed that some genetic material is obtained by R strain from heat-killed S strain and it changed genetically and morphologically to S strain. Martha Chase and Alfred Hershey worked with both E. coli and bacteriophage by using P32 and S35 isotopes and established that it was DNA of virus that entered the bacteria but not protein. Coming to the second question. Choose the correct expression. You have to choose the correct expression with respect to experiments done by Frederick Griffith using R and S strains of Streptococcus pneumoniae. Living R strain of Streptococcus injected into mice. Mice died. So how they will die? R strain is a virulent strain, doesn't cause any disease. So mice survived. So this is not the correct option. Living S strain of Streptococcus injected into mice. Mice remained alive. No. S strain is virulent. When injected into mice, so naturally they die. So they died or they were killed. This is also not correct. Heat killed R strain together with living S strain injected into mice. Mice survived. How they survived? This S strain was not heat killed. Only R strain. The strain is virulent. Naturally, they cannot survive. So that's why they could not survive. Right? So this is also incorrect. They must be killed. Heat killed S strain together with the living R strain injected into mice. Mice died. So naturally, so when heat killed S strain together with the living R strain injected into mice, living R strain obtained some chemical substance from heat killed S strain and transformed into S strain, and the mice developed pneumonia symptoms and died. The fourth option must be the correct one. Let us see the answer. Fourth one. R strain of pneumococcus is virulent and S strain is virulent. When Griffith injected living R strain and heat killed S strain separately into mice, mice survived. If heat killed R strain and living S strain together injected, the mice would die as the virulent strain is living. When he injected heat killed S strain along with the living R strain, the mice were killed because some chemical was obtained by living R strain from heat killed S strain and it transformed into S strain, and hence the mice were killed due to pneumonia. Coming to the third question. Statements. Let identify which statement is correct, which statement is incorrect, or whether both are correct or incorrect. Transformation in bacteria was discovered by Frederick Griffith. Absolutely true. Griffith discovered that it was a DNA that was obtained by R strain from heat killed S strain and transformed into S strain. So Griffith could not discover whether it was DNA or RNA or protein. He could not establish, but they thought of it must be the protein, not DNA. So statement 2 is wrong, statement 1 is correct, but statement 2 is incorrect. First option must be the correct one. Let us see the answer. Transformation of R strain of Streptococcus pneumoniae into S strain was discovered by Frederick Griffith, but he didn't discover DNA, the substance that was obtained by R strain from killed S strain and transformed as S strain. On to the next question. Which one is not a feature of S strain of Streptococcus pneumoniae? Should not be the feature of S strain of Streptococcus pneumoniae. S means virulent strain. The smooth and shiny colonies it forms. It forms smooth, shiny colonies when grown on culture plates. This is feature of S strain. It has a mucus coat around its cells as it has got capsule. It causes pneumonia disease in mice. This is also correct one. So these first 1, 2, 3 are the features of S strain. Actually, fourth one must be the not the feature. If it is heat killed, it causes pneumonia in mice and mice are eventually killed. No. When heat killed, they survive. Mice survive. So fourth option must be the not a feature of S strain of Streptococcus pneumoniae. Only living S strain can cause pneumonia and eventually the mice will be killed. So fourth option must be the correct one. Heat killed S strain of Streptococcus pneumoniae doesn't cause any disease in mice. Coming to the next question. The assertion and reason type. So you have to conclude whether assertion and reason are correct, or one is correct, other one is incorrect. If correct reason is correct, whether explaining the assertion or not, that you have to decide. Biochemical nature of transforming principle in pneumococcus was established by Avery et al. Absolutely true. Avery and all purified the biochemicals, proteins, DNA and RNA of R strain of pneumococcus. So they have purified the biochemicals of proteins, DNA and RNA of S strain of pneumococcus. The assertion is correct, the reason is wrong. The first option must be the correct one. Let us see the answer. First one. Biochemical nature of transforming principle was established by Avery et al. They purified the biochemicals, protein, DNA and RNA of S strain of pneumococcus, not R strain, S strain only they purified. They showed transformation when the proteins were digested by proteases and RNA by RNases. Digestion of DNA with the DNases could inhibit transformation, and it showed DNA of S strain as a transforming principle that transformed R strain into S strain. They concluded DNA as the hereditary material. Coming to the next question. In Hershey and Chase experiment, the protein coat or capsid of bacteriophages was labeled with the radioactive isotope. So proteins have got sulfur, they do not have phosphorus. They use the sulfur as isotope S35. S35, not S32. They used P32 to label the DNA of the bacteriophage. They have not used P35. S32, P35 were not used. S35 and P32 were used. S35 was used to label the protein coat and P32 label the DNA of the bacteriophage. So S35 must be the correct one. Third option must be the correct one. The protein coat of bacteriophage was labeled with S35 as the proteins have sulfur as a component of sulfur containing amino acids like methionine and cysteine. Proteins do not have phosphorus. And the nucleic acids are DNA doesn't have sulfur. DNA of bacteriophage was labeled with P32 isotope because DNA has phosphorus as part of its nucleotides. They have not used S32 and P35 isotopes. Coming to the next question. Identify the correct statements with respect to Hershey and Chase experiments. Hershey and Chase subjected the bacteria infected with labeled bacteriophages to blending to remove the viral protein coats from bacteria. Absolutely true. They subjected them to blending, kept in blender and they did blending. So that the ghosts of the bacteriophages or protein coats of bacteria can be detached from the bacteria. Hershey and Chase separated the viral particles from bacteria by centrifugation. So they carried out centrifugation and separated. Right? They have got supernatant and also the sediment they have got. Bacteria infected by viruses having radioactive DNA were radioactive. Bacteria infected by viruses having radioactive DNA were radioactive. This is also true. They have shown radioactivity, the bacteria which were infected by viruses having radioactive DNA. But bacteria infected by viruses having radioactive protein were not radioactive. Bacteria infected by viruses having radioactive protein were radioactive. No, this is wrong. This is wrong. Upon blending and centrifugation of bacteria infected with viruses with radioactive protein, the supernatant showed radioactivity. Yes, it has shown. So radioactive protein will be there in the supernatant. It shows radioactivity. So the bacteria are at the right. So A, B, C, and E are correct. A, B, C, and E. Second option must be the correct one. Second. Here is the explanation correct. See here, the first bacteriophagous labeled with S35. S35 could label the protein capsule. So the second one, P32 isotope was used that can be incorporated into the DNA of the bacteriophagous. So when infection is done, both infect the bacteria and after infection, successful incorporation of the DNA into the bacteria, blending is done. In both the cases, the viruses, the bacteria, the ghosts are separated from the bacteria which are infected. So after that, they are subjected to centrifugation. So, in subjected to centrifugation, the bacterial cells, no radioactivity was detected because sulfur is not an integral part of nucleic acids. So, radioactivity is detected in the supernatant which has got protein codes of the viruses. So, in this case, in the second case, radioactive P32 detected in the cells, bacterial cells. So, radioactivity was not detected in the supernatant where the viral capsids are present. So, this is the experiment carried out by Hershey and Chase. So, this is very clear. Coming to the next person. The genetic material in QB bacteriophage is not QB, it should be Q beta bacteriophage is. So in the book, it was printed as B, actually it is beta, Q bacteriophage. Q beta bacteriophage is. Q beta bacteriophage, some bacteriophages have got RNA genetic material. Here it is single stranded RNA. It is a plus strand RNA. Plus strand RNA can directly code for proteins without forming the corresponding messenger RNA. Such RNAs are plus strand. Minus strand, from RNA there is the formation of another RNA, that is messenger RNA, and then it codes for the protein. So it is single stranded RNA. Genetic material in Q beta bacteriophage is. So single stranded RNA, fourth option must be the correct one. Let us see the answer. Fourth one. Some bacteria like Q beta have plus strand ssRNA as its genetic material. Such RNA is directly involved in translation in host bacterial cell. Coming to the next question. Choose the incorrect statement. Both RNA and DNA are able to generate their replica. This is the correct statement, but you have to choose incorrect one. Both RNA and DNA structurally and chemically more stable. No. Only DNA is more stable, RNA is less stable because RNA is single stranded. No nitrogen based complementarity usually, only at certain places it may be. Moreover, ribose has got highly reactive OH group at the second carbon, and uracil is less stable than thymine of DNA. Right? So, chemically more stable. Now, only DNA is more stable than RNA. Both RNA and DNA are able to mutate, absolutely true. But RNA can mutate faster because it's highly reactive. DNA slowly it is subjected to change or mutation. RNA is able to express itself in the form of Mendelian characters. Yes, RNA can be directly expressed to form the protein, but DNA, unless it is transcribed to messenger RNA, and messenger RNA later it is expressed. So RNA is able to express itself in the form of Mendelian characters. So this is correct statement, but incorrect one is both RNA and DNA structurally and chemically more stable. This is incorrect statement. RNA is less stable, DNA is more stable. Let us see the answer. Second, DNA can generate its replica with the catalytic activity of RNA-dependent RNA polymerase and DNA by DNA polymerase. Structurally and chemically, DNA is more stable than RNA, as the DNA is usually double-stranded. It is not having reactive OH group at second carbon of sugar, and it has stable thymine instead of relatively unstable uracil. Both DNA and RNA are able to mutate, but being more reactive, RNA mutates faster than DNA. RNA can easily express Mendelian characters as it can directly code for proteins. DNA has to be transcribed into mRNA before coding for proteins. Coming to the next question. Choose the incorrect statement. DNA was the first genetic material. No, RNA was the first genetic material. This is the incorrect one. RNAs, since usually single-stranded, cannot repair the changes occurred in it during its formation. Yes, there is no repair process of RNA, being single-stranded usually. Essential life process, metabolism, translation, and splicing evolved around RNA. Yes, this is also correct statement. So second correct, third one is also correct statement. Some biochemical reactions are catalyzed by RNA. Absolutely true. Some biochemical reactions are catalyzed by RNA, like peptidyl transferase, which has got 23S ribosomal RNA. So RNA molecules can act as enzymes or biocatalysts, but not DNA. So the correct statement is the first one. Let us see the answer. Solution. RNA was the first genetic material because metabolism, translation, splicing were first evolved around RNA, as it can directly involved in coding for proteins. Eventually, DNA has evolved from RNA after certain modifications it needed for stability, such as becoming double-stranded. Ribose sugar changed to deoxyribose with the removal of oxygen at second carbon of sugar present as OH. Uracil changed to thymine by methylation. So please like, comment, subscribe. Thank you.